Hi everyone, this class is about protein metabolism. In this class, we'll see about the digestion and absorption of a protein and what is urea cycle and the disorders and regulation of a urea cycle. So before going to the metabolism of protein, we'll just see the introduction of protein and amino acids. The proteins are the primarily constituents of the body which may be structural or functional. A regular and adequate supply of protein in diet is essential for cell integrity and functions of a cell. So these proteins are made up of amino acids which are essential to life, have a role in metabolism and are important in nutrition. So these amino acids form short polymer chains called peptides as well as longer chain that are called polypeptides or proteins. About 75% of the human body it's made up of chains of amino acids which is so vital to our system functions, and all the chemical reactions that occur in the body depends on amino acids and proteins they build. So the primary use of amino acids provided through the degradation or digestion is as building blocks for the synthesis of proteins and other nitrogenous compounds such as nucleotide bases. And these amino acids in excess of those needed for synthesis can neither be stored in contrast with fatty acids and glucose nor excreted. Rather, surplus amino acids are used as metabolic fuel. So of the 20 amino acids commonly found in proteins, that are 10 are not essential in the diet as they can be synthesized by the body. So here we can see the basic structure of amino acids, which are derivatives of carboxylic acids formed by the substitution of alpha hydrogen for amino functional group. So the carboxylic functional group at the one end and amino group at the other end, and also side chain is present and attached to the alpha carbon atom. So this forms the basic structure of a amino acid. And once we intake the proteins to our body, it undergoes degradation by enzymes as proteases. So it includes exopeptidases and endopeptidases. Endopeptidases, they act on peptide bonds inside the protein molecule so that the protein becomes smaller and smaller units. This group includes pepsin, trypsin, chymotrypsin and elastase as enzyme, and exopeptidase which act only on the peptide bond located at the ends of the polypeptide chain. So this grouping includes carboxypeptidase which act on peptide bond at the carboxy terminal end of the chain, and aminopeptidases acts only on the peptide bond at the amino terminal end of the chain. So these proteolytic enzymes break down dietary proteins into their constituent amino acids. These enzymes are produced by three different organs as the stomach by the action of HCl and pepsin, and in pancreas trypsin, chymotrypsin and elastase is present. In intestine, carboxypeptidase, aminopeptidase and dipeptidase. So these are the enzymes which are used in the digestion of proteins to amino acids. So as we know, there is no digestion of a protein in mouth, it starts in stomach. So when protein enters the stomach, it stimulates the secretion of the hormone gastrin from gastric mucosal cells, which in turn stimulates the release of gastric juice containing hydrochloric acid, that is HCl, and the proenzyme pepsinogen and renin in infants. So the HCl it makes the pH optimum for the action of pepsin and also activates the pepsin and denatures the proteins. So here the renin, otherwise also called as chymosin, which is only active in infants, not in adults, and this milk protein casein is converted to paracasein by this action of renin. And the pepsin it's secreted by the chief cells of stomach as inactive pepsinogen, later which is converted to pepsin as an active form by the HCl. So the conversion of pepsinogen to pepsin is brought about by removal of 44 amino acids from the N-terminal end by the HCl. So the optimum pH will be around 2. The pepsin is an endopeptidase. The pepsin catalyzes hydrolysis of bonds formed by carboxyl group of phenylalanine, tyrosine, tryptophan and methionine. By the action of pepsin, these proteins are broken down into proteoses and peptones. So the pancreatic digestion of proteins. The optimum pH for the activity of a pancreatic enzymes is 8. It's provided by the alkaline, bile and pancreatic juice. The secretion of pancreatic juice is stimulated by the peptide hormones, that is cholecystokinin and pancreozymin. This pancreatic juice contains the important endopeptidases, namely trypsin, chymotrypsin, elastase and carboxypeptidase, which acts on the proteins to form amino acid. These enzymes are also secreted as zymogens, that is in active form as trypsinogen, chymotrypsinogen and proelastase. And all the three are serine proteases, that is the active centers of these enzymes contain serine residues. Next is trypsin. Trypsinogen is activated by enterokinase present on the intestinal microvillus membrane. So once trypsin is activated, it activates other enzyme molecules also. Trypsin is activated by the removal of a hexapeptide from N-terminal end. So this catalyzes hydrolysis of the bonds formed by carboxyl groups of arginine and lysine. And acute pancreatitis, it's a premature activation of a trypsinogen inside the pancreas itself. So it will result in the autodigestion of pancreatic cells. And the chymotrypsin, the trypsin will act on chymotrypsinogen in such a manner that A, B, C peptide bonds are formed. These three segments are approximated so that the active site is formed. So it converts chymotrypsinogen to chymotrypsin. Thus, selective proteolysis produces the catalytic site. And the carboxypeptidase, the trypsin and chymotrypsin degrades the proteins into small peptides, and these small peptides are further hydrolyzed into dipeptides by the action of carboxypeptidase, which is present in pancreatic juice. The procarboxypeptidase is activated also by the trypsin. And further, the digestion of proteins in intestine, that is complete digestion of a small peptides to the level of amino acids brought about by enzymes present in the intestinal juice by succus entericus. And the luminal surface of intestinal epithelial cells contain the following enzymes as aminopeptidase, which releases N-terminal amino acids, and dipeptidase and tripeptidase will complete the digestion of a protein. So once the protein is taken to our body, it gets digested and absorbed by intestine and passed to the portal hepatic vein. So these amino acids once get hydrolyzed by the enzymes, these amino acids occur mainly in the small intestine. It's an energy requiring process, and these transport systems are carrier mediator or ATP sodium dependent import systems. So here the L amino acids are selectively transported across the intestinal mucosa, which need carrier like sodium pump or sodium ions and also ATP molecule. So several sodium dependent transport proteins are found in the brush border membrane, that is not specific for each amino acid but rather for the groups of structurally similar amino acids. And the amino acids are transported by passive diffusion, where the ATP molecule is not required, and thus amino acids are released by digestion and pass from guts to the hepatic portal vein to liver. So here the proteins are constantly being synthesized and then degraded. So once it is done, the catabolism of a amino acid starts, as it includes transamination, deamination, disposal of ammonia. So the end product of the protein metabolism will be ammonia, and it is transported to liver. So for the absorption of these amino acids, different carriers will be there as neutral amino acids. For basic amino acids there will be different amino acids and acidic and beta amino acids. In intestine, kidney tubules and brain, absorption of a neutral amino acid is affected by these cycle as a gamma glutamyl cycle. So we'll know about the glutathione. It's a tripeptide with the amino acids, forms a gamma glutamyl amino acid that is then cleaved to the free amino acids, which are net result in the transfer of amino acid across the membrane. So in this cycle also the amino acids will transfer. So once the ammonia is formed, it has to be disposed by the body. So the complete catabolism of amino acid includes 3 stages: transamination, deamination and disposal of ammonia. So here this ammonia gets converted to urea by the urea cycle. So the ammonia is produced in most tissues. Since ammonia is extremely toxic, it is immediately converted to non-toxic metabolites such as glutamate, glutamine, alanine, and ultimately to the urea. So here the urea cycle plays a major role. The sequence of reactions involved in the biosynthesis of a urea, which are summarized as a few steps. So urea is a major end product of the nitrogen metabolism in humans, and also the significance of urea cycle includes the toxic ammonia gets converted to harmless non-toxic urea. It disposes of 2 waste products as ammonia and carbon dioxide and forms semi-essential amino acid as arginine and participate in the regulation of a blood pH. So this is the urea cycle. It has 5 steps. We'll see one by one. As first, the formation of a carbamyl phosphate. Here the biosynthesis of urea begins with the condensation of carbon dioxide, ammonia and ATP, which forms carbamyl phosphate. A reaction catalyzed by mitochondrial CPS1, that is carbamyl phosphate synthetase. So the formation of carbamyl phosphate requires 2 molecules of ATP. One ATP serves as a source of phosphate and second is converted to AMP and pyrophosphate. So once the carbamyl phosphate forms, it leads to the formation of a citrulline, that is a second step. As the carbamyl phosphate donates its group to ornithine, it forms citrulline and release phosphate, which is catalyzed by ornithine carbamylase. So formed citrulline passes to cytosol of a liver cell from the mitochondria. And the next step, the formation of arginosuccinate. The transfer of the second amino group to citrulline occurs by a condensation reaction between the amino group of aspartate and citrulline in the presence of ATP to form arginosuccinate. So ATP is cleaved into AMP and phosphate. This reaction is catalyzed by arginosuccinate synthetase of the liver cytosol. So it's a magnesium dependent enzyme. And the next, the formation of arginine and fumarate. So from the arginosuccinate, arginine and fumarate is formed by the arginosuccinate lyase or arginosuccinase enzyme. Here the arginosuccinate, it forms a free arginine and fumarate. The fumarate so formed returns to the pool of a TCA cycle intermediates, and through fumarate urea cycle is linked with the citric acid cycle. The two Krebs cycles together have been referred to as Krebs bicycle. So the last step of urea cycle will be the formation of urea and ornithine. So in the last reaction of a urea cycle, the liver hydrolytic enzyme arginase cleaves arginine to yield urea and ornithine. Ornithine is thus regenerated and can enter mitochondria again to initiate another round of a urea cycle. So here the arginine cleaves to yield urea and arginine by arginase enzyme, and once ornithine enters into mitochondria, it initiates another cycle. So thus urea is formed and excreted in the urine. Thus human beings excrete about 10 kg of urea per year. So as the cycle involves energy cost, here the overall reaction may summarized as ammonia plus carbon dioxide and aspartate, which leads to the urea and fumarate formation. The urea cycle consumes 4 high energy phosphate bonds. 2 are used in the first reaction needed to make carbamyl phosphate, and 1 is required to make arginosuccinate, and 1 is required to restore AMP to ADP. The urea cycle and TCA cycle are interlinked, and so it's called as urea bicycle. And once the urea cycle completes, we'll see the regulation of a urea cycle, which includes substrate availability. That is, higher the rate of ammonia, the formation of a urea cycle will be in higher rate. And allosteric regulatory enzyme, which includes its a major regulatory enzyme, is CPS1, that is carbamyl phosphate synthase 1. It's stimulated by the N-acetyl glutamate, and arginine is an activator of a N-acetyl glutamate synthase, and also regulated by the compartmentalization. That is, the urea cycle enzymes are located in such a way that the first 2 enzymes are in mitochondrial matrix. So first 2 reactions will take place in the mitochondrial matrix, and the inhibitory effect of fumarate on its own formation is minimized because arginosuccinate lyase is in the cytoplasm. So while fumarase is in mitochondria. So these are the regulatory enzymes of a urea cycle. And we'll come to the disorders of urea cycle. The deficiency of any of the urea cycle enzymes would result in hyperammonia, because ammonia has to convert it to the urea. So when the block is in one of the earlier steps, the condition is more severe, since ammonia itself gets accumulated, and deficiencies of other enzymes result in the accumulation of other intermediates which are less toxic, and hence symptoms are less. So compared to the ammonia, other intermediates effects are less. And as a general description, disorders of urea cycle are characterized by hyperammonia, encephalopathy, and respiratory alkalosis. Clinical symptoms include vomiting, irritability, lethargy, and severe mental retardation. Infants appear normal at birth, but within days progressive lethargy sets in. So mainly it affects to brain, as brain is very sensitive to ammonia. Child may be put on low protein diet and frequent small feeds are given to child. And since citrulline is present in significant quantities in milk, breast milk is to be avoided in citrullemia. And ornithine transcarbamylase deficiency is the only urea cycle disorder which is inherited as an X-linked trait. And elevated blood levels of ammonia are associated with high glutamine levels in CSF and blood. So arginosuccinate lyase deficiency leads to the arginosuccinic acidemia, and therefore metabolic acidosis also occurs. And hyperammonia is less severe, and arginosuccinate is elevated in CSF and excreted in the urine. So here in the table we can see the urea cycle disorders as first diseases and enzyme deficient and features. So diseases as such, hyperammonia type 1, the enzyme deficiency will be CPS1. And the features, it's a very high ammonia levels in blood, autosomal recessive, and also cause mental retardation. And the hyperammonia type 2, the enzyme deficient will be OTC, that is ornithine transcarbamylase. The ammonia level high in the blood and increased glutamine in blood, CSF and urine, orotic aciduria due to channeling of the carbamyl phosphate into pyrimidine synthesis X-linked. And the next, hyperornithemia, that is defective ornithine transporter protein. So failure to import ornithine from cytoplasm to mitochondria, defect in ornithine 1 gene. And the next, citrullinemia, that is enzyme deficiency will be arginosuccinate synthetase, and the features will be autosomal recessive in inheritance, high blood levels of ammonia and citrulline is absorbed. Arginosuccinic aciduria, that is enzyme deficient will be arginosuccinate lyase. The arginosuccinate in blood and urine will be more, and the hyperarginemia, as the enzyme deficiency will be arginase. The features includes arginine increased in blood and CSF. Instead of arginine, cysteine and lysine are lost in the urine. So this includes the metabolism of protein part as digestion and absorption of a protein, and the catabolism of a protein as one of the part, urea cycle, the regulation and disorders of a urea cycle. So in next class we'll see further part of the protein cycle. Thank you.